Thank you, Dr. Palok, for inviting me, and I thank my fellow Bankos people there. So so thanks Bankos for also hosting me here. I would be talking. My team focuses on undernutrition, biology, and microbiome, and particularly on iron deficient anemia. This talk is directed on what we want to do and what we have achieved in the last couple of years on iron deficient anemia. So we have this burden of about 2 billion population across the world has iron deficient anemia, and about most of these are in the early life of starting from very early in the childhood to all the way to the woman in reproductive age and goes on to most of the crucial years like during pregnancy and so on, and then it actually throws the next generation into a vicious cycle of undernutrition. So, which means there is this 2 billion population, and then they expose the next generation into this vicious cycle of undernutrition. So, that's the kind of burden we are looking at. And what do we do for iron deficient anemia? What we do is just pump in more iron for this patient. So that's the only strategy we have to treat this iron deficient people. I'm sure most of us, the senior people would have had black colored tablet in schools those days, versus I don't know the younger generation what they get in schools for medical, there will be medical screening and then they give this black colored tablets, and these are iron tablets. So, and for the last 30 decades, that's all we have been doing for treating anemia, where we just give more iron as supplement for iron deficient anemia. So if that were to work, our burden of anemia, they've given me, two things to juggle with, so I'm little, not getting used to this. So if you see this, there is about 67% of our population in our country are still anemic despite iron supplementation for 3 decades. So which means if that were to be the treatment, we should be having anemia or the Anemia Mukt Bharat by now, right? We don't have an Anemia Mukt Bharat yet, mostly because just supplementing empirically iron doesn't help iron deficient anemia. Okay, so what is that we are missing? That's the starting point 10 years ago, and then we're still working on it. What we found is that I particularly work on the microbiota gut nutrient axis, where we try and understand what kind of microbes have changed due to this iron deficient anemia, and how we're going to treat this effectively iron deficient anemia. So some ideas that we considered before we started. One is from the soil biologist that we took. If you see, there is this 2 abiotic factors which we generally don't consider, doctors don't consider. So the redox potential and the pH of a niche. Okay, the soil biologists actually understand this very well. They understand the pH and the redox of a particular niche, which actually determines the growth of a plant. So they use fertilizers, the biofertilizers nowadays, or the organic farming. These are some catch terms, but what they essentially do is they reset redox potential for making the conditions favorable for the plant growth. So the plant growth is not just growing plants, but also in terms of the yield. To increase productivity, they try and reset all this abiotic factors, and that's one of the ways that the farmers, they get better yields. So can we apply this for a disease condition, particularly like iron deficient anemia, where can we look at favorable conditions for these nutrients to be absorbed? That's the question we had as we begin with. And then we know that there are multiple factors that could actually enhance iron absorption. And as a biochemist, I teach medical students saying that iron can be regulated only at the level of absorption. There is no excretion for iron. So we tell them that there is this mucosal block theory. Iron has to be regulated at the level of absorption. So that is all we have, because iron is tightly regulated in the body. We can't throw iron because it's too precious. So we only know and want to try and understand what are the factors that will actually essentially make the iron bioavailable so that our cells can take it. Well, starting from short chain fatty acid, the phytase that the microbes can produce, and the various microbes that can produce heterophores and so on, to take the extra iron, are possibly options that are there in the milieu where iron gets absorbed. Okay, well, so these are some of the publication and experiments that we have done. One, where we have actually fed this girl with chocolate chip cookie, and this cookie has prebiotics. A prebiotic is nothing but complex carbohydrate. When we can't digest it, it goes all the way to the colon, and then it promotes microbes to eat it up, and then the microbes flourish in the colon, and then this in turn helps. What you see here is that the pH, acetate, butyrate, or some of the short chain fatty acid that keeps increasing in concentration as they start taking this across various time points when we feed them. And then the pH also changes, the fecal pH reduces, and then we have seen there is this zinc absorption is increasing in this patient. This is published where the zinc absorption part is not done here because that is radio labeled zinc was fed. This was done in Africa, in Malawi. So the first part was done in our country, where we tried to change the microbiome using prebiotic feed. Well, so essentially we changed the pH and SCFA production. So now we are thinking, what are the common foods that we have across our country, and then we found that the spices, fermented food, and high fiber, these are some options we have where we could increase this resistant starch content or the prebiotic in the food. Well, before that, before designing what could be the exact food, we wanted to have some biomarkers that can predict actually whether this person is anemic, and can we reset to this person to non-anemic. We just took top 20 microbes in anemic individual with just hemoglobin and neutrophil lymphocyte ratio. Neutrophil lymphocytes these are the basic tests that one does in a peripheral setup, where hemoglobin is measured, and then they measure a differential count where they can measure the neutrophil percentage and the lymphocyte percentage. So what does it give? Neutrophil gives acute inflammatory condition, lymphocyte chronicity of the inflammation. So we just took a simple ratio of neutrophil versus lymphocyte, and then we measured hemoglobin level in the top 20 microbes. Of course, microbes we are not going to do in the field to measure or look for anemia. This is just, we developed a predictive tool to design our food. And then we found that we could with more than 80% accuracy, we can predict the disease anemia versus non-anemia. And we also identified a particular microbe called Roseberia faecis, which is more predictive in particularly low NLR population. And one more interesting microbe that we identified was a segmented filamentous bacteria. We found, and all the blue spokes that you see here are from India. Rest of them are from rest of the world, and then we collaborated with a French partner, and then we looked at our SFBs versus the SFBs that they have characterized across the world, and then this is where we stand from our anemic population. We also see that if the SFB concentration were to increase in anemic population, it increased with the neutrophil lymphocyte ratio also increasing. So which means the neutrophil concentration is increasing with the increased SFB. So which is again, there is some acute event that is happening all the time. So then we had to go to the site of where the actual action happens. That's the duodenum. where the iron absorption actually happens. So we took the biopsy from the duodenum. We looked at microbiome difference at the duodenal site, and then we found that with respect to the neutrophil and lymphocyte ratio, we found a lot of these microbes that are varying. So which means there is microbial association with respect to inflammation happening. So we, one step ahead, we decided we look at the microbiome and the host interaction. What we found is that the redox, in terms of the MRI we had published earlier, where microbial redox index we derived, and then with respect to, if you see, there are these two lines which are similar, whereas these two are different, where this one is an anemia, where the anemic individual lost the balance between this redox microbes, which was the ratio between the aerobes and anaerobes. The anaerobes were all over the place, while in normal individuals the anaerobes were not that high and duodenum, which is expected. So we looked at the microbes and all the genes that were upregulated and downregulated. In controls, there is a pattern that you see here. There is, if you see, there is left-right interaction that is happening between the redox group of microbes, while in anemic there is an up and down shift, which means the response to the host microbe interaction is totally different in anemic versus non-anemic. So then we got a little more curious, looked at what exactly is happening, because this could now be more of a mucosal disorder that we are dealing with rather than anything else. So we looked at the three layers that we looked at. One is the blood test that we do. Any lab you go, the blood test is done. We looked at the neutrophils, monocytes, all the iron parameters and neutrophils and so on. And the middle portion is the microbiome that is there in the duodenum, not in the stool. While the pink layer that you see here is the gene that upregulated or downregulated in these anemic individuals. So we see that there is a tight package here where there is all these iron parameters in the blood are associated with microbes here, and this particularly is associating with one of those RNA that is a non-coding RNA that is not described yet, that is there. So there is a lot of work now for us where we want to understand what is this RNA doing, and then we have to explore this further. Meanwhile, what we have is that we know now we have to modify a lot of the niche, particularly at the duodenal level, and then the one way that to do is to measure, intervene it using various options that we have here. And is that the mic? Oh. So, okay. I thought I was running out of time, so I'll take just 5 more minutes. So, one example I have where we used food, basically, of the options that I showed, we just chose the dietary intervention, particularly with the prebiotic and symbiotic, where one of the population that we thought we will, we thought we should intervene is that the pregnant women, because pregnant women, the national program says all the pregnant women in our country, irrespective of whether or not they are anemic, should get iron supplement. About 60 milligrams are pumped into them. So this is a cohort where they are given as a national program, iron has to be supplemented no matter whether they are anemic or non-anemic. So what we did, we went to them, we gave one group of pregnant women, iron has to be given because I have no control on it, it's a national program. So what I have control is, I just gave one group a prebiotic shake. Let me call it Nioba, native yogurt banana shake. Native yogurt banana shake. Okay, we just gave them a food as an intervention for 6 months, 180 days of iron supplementation along with this shake, and then what we achieved was this. has is nothing but a height for age z-score, where we look at the height in the newborns. We looked at the newborns, we gave the mothers, we followed them at various time points up till 1 year of the child's life. So at the end of the first trimester, we recruited them. We started giving them from, that is the national program, because starting of the second trimester onwards, the iron supplementation goes in, and then along with the iron supplement, we kept giving them the shake, and every month we started collecting samples, and then we measured all kinds of parameters that I will explain now. So this is what we found. There is it's 0.056. Forget significance. There is a change in height between intervention and control group. And we looked at the microbiome change. Yes, there are microbiome changes in microbes. So then we were curious what these microbes were doing. What we found is that iron redox capacity, we looked at iron acquisition and storage and iron reduction. These are three parameters in terms of iron we looked, and then we found that the iron acquisition was significantly improved with the intervention at the second trimester and the third trimester, while this iron acquisition had increased the reduction capacity, or iron was reduced more at the intervention group. And we found these are the microbes that were responsible for the changes that you see here. Okay, well, so this is in the mothers, and now we want to look at what is happening in the infants. Only at the day 7 there was a change, and then we found that there is a lot of these microbes were responsible for this change that in terms of microbes. But then that's not what we were interested in. We are interested in the outcome on the development of the child. So we looked at how this microbiome was maturing in the children across time point. Say the time points were 7 days, 6 months, and 1 year. And we measured the microbiome pace, and then we looked at the microbiome pace where there is this intervention group was slightly better than the control group. We saw that intervention group had 2 spikes. We know that the microbes that are responsible for the 2 spikes as opposed to a diffused spike in the control group. And then finally they were all catching up towards the end, but then were they the same. So when we looked for more in terms of the height for age, then we found that the intervention group had a significant positive trend across time. And then at 6 months particularly, the line is crisscrossing, and then it's a positive trend in terms of height and the microbiome maturation. So which means the 6 months is a time when a baby is exclusively breastfed. The mother's breast milk is the only food that the baby gets for 6 months. So we were now curious, is the breast milk has something to do with the maturation? Because after this, till this 1 year point is the food that we feed from outside. That's the next part of the story, which we got a grant explaining that, we have to do a complementary feeding, and we are now developing what is called a take-home ration, a microbiota-directed complementary feeding, where we are developing a take-home rations, and that's a project, that's an ongoing project, which to take care of the difference between 6 months to 1 year. While now we have something happening between 7th day and 6 months, and then we looked at the breast milk microbiome and how it interacts with the fecal microbiome in the child at 6 months. And then you see here, this one is the intervention child, where you see there's a lot of crisscross happening between the breast milk microbiome and the fecal microbiome of the child, while here this is the control, where there are these 3 big spots standing separately. Okay, they are not crisscrossing. What does this crisscrossing mean? I mean, my earlier speaker, he was talking about how there is bacterial biofilm, multiple organisms together, and how they interact, how they promote each other, and so on. And that's possibly happening here, where there is this more the networking, the better the niche in the intestine, and then better health of the baby. So this is an ongoing study, and that's all I have for now. I'll be happy to take more questions. And yeah, so form the gut, or we feed the microbes with various options that we have. We are even starting fecal transplant in our institute on autistic children. So these are some things that is happening in our, and I thank my team and my mentor for all the work that is happening in our team. Thank you so much. Thank you, Dr. Palok, for